All right, so this is a really short video. We're going to look at three cells that we want to know, and we're going to look at three forms of cell locomotion, all right, and kind of how cells get around. So this first video, all right, we're going to look at Paramecium. All right, so that is the name of the organism. We want to know this organism, okay? And they get around using cilia. Cilia are these little teeny tiny hair-like extensions. They're on the outside of the cell, and they are just kind of used to beat against the water, beat against that fluid. Think of them kind of like oars. So they all go in one direction, then they help the paramecium get around. So we'll look at them for a minute. You can just see they kind of flip back and forth, and they propel the paramecium through the water. A couple other things about the paramecium. You can see right here, that little, let me go back and pause it. If you look right there at the bottom, that's called the oral groove. It's kind of like the mouth of this cell. So that's the paramecium. And those are the cilia. You can see that this is definitely a eukaryote. It has organelles visible in the cell. All right, now, euglena, this is a little longer video, so if you want to stick around and watch it, you can. But a euglena is this green single-celled organism here. It does conduct photosynthesis. It also has an organelle called an eye spot, which can sense light. And that's obviously important for an organism that does photosynthesis, right? It wants to move around and find a good place to produce light with its little green organisms there, organelles there. You can see the flagellum is a really good way to move around. These guys are moving pretty quick. And so this is at 40x magnification. They're going to zoom in a little bit to 100 and then go a little higher. And we'll be able to see that flagellum in greater detail. Now, basically, a flagellum is like a whip, okay, or a tail. And it kind of spins and slaps against the extracellular fluid, the water, whatever it's in. And that's one of the ways that the euglena gets around. And euglena are pretty interesting little creatures. This is called a dark field microscope. It's similar to a light microscope, except obviously it has a dark field. And it allows a little bit greater, in my opinion, contrast for some structures. So you can see a little bit more of what's going on. So this is 600x zoomed in, and maybe we'll get to see a flagellum here in just a second. You can kind of see one there on the right. Let's see. There it is, kind of moving through extracellular fluid there. This was kind of bogged down in some kind of detritus, some kind of debris. Here you can see the whip-like tail. It actually has two flagellum on the euglena. There's the eye spot we talked about a moment ago. It's got a pigment in there that detects light. And hopefully down here at the bottom, see, we'll get lucky. We can see one of these. Yep, there we go. One of those flagellum. All right. The last one is the amoeba. And amoeba gets around using pseudopodia or pseudopods. Now, pseudo means false. Pod is the suffix that means foot or feet. So a pseudopod is a false foot. And you can see the cytoplasm in this amoeba. This is, again, a dark field microscope, so you can see those organelles really well. The cytoplasm is pushing the cell membrane out to form these feet, these little false feet, these little projections. And so that allows the amoeba to kind of wrap itself around prey, to engulf the prey, and lets it move around its environment. So those are the three kinds of single-cell locomotion. Right, you have pseudopods, which are false feet, flagellum, which are whip-like tails, and cilia, which are little hair-like projections. We also want to know the paramecium, the euglena, and the amoeba. We want to know a little bit about each of those, okay? So, short video. That's all we've got for this one.